Okay, Mazi, you're free.
Okay. Do you see my screen?
Yes.
Okay, perfect. Shall I start?
Please go ahead.
Okay. Well, first of all, thank you so much, Jay, for the invitation. I tried as much as possible because I was teaching on Thursday mornings, so I couldn't attend all the journal clubs myself, but the ones that I attended, I learned quite a lot. I wanted to use the opportunity, since I'm new in town, to tell you what we do in general in my lab. So I joined IOP on January. So we're making an experimental lab with the two main focus on hydrodynamics and biophysics. And in the hydrodynamics course, we work on active and ultra soft matters. When I call ultra soft matters, I mean materials that can barely resist their own weight. They're super soft, in fact, and also a lot of classical fluid mechanics. And I call it classics because there's now, with active matter, soft matter, there's a new wave of fluid mechanics. And most of those works are a combo of experiments and theory. Most of actually my theoretical works are on hydrodynamics. We also do biophysics that is related to mechanosensing, light sensing, and motility and non-motility of some living organisms. I talk about that, in fact, today. So I decided, when Jay asked me to give a talk, I kind of struggled what topic to talk. And I found most of the audience are theoreticians here. So I decided to talk about something that is not actually include much theory. So for the sake of diversity of the topic. So I'm going to talk mostly about biophysics. And the topic I chose is around bioluminescence in unicellular organisms. The paintings I have here is from Joseph White. It's about the medieval times when alchemy was the things, and philosophers or alchemists started looking at philosopher's stone. And I will, in the middle of my talk, I will tell you why I have this beautiful painting that I love personally as my starting title slide. So the content is like this. I will talk a little bit about basics. Then I will review the literature, and I do it in a very unconventional way, which instead of talking about literature as technical literature, I will more talk about history, in fact, and I go really back in time. And then I'll show you what we've done around this historic, actually, research topics and what we plan to do. So I keep it very high level. I don't talk about much technicality, except in some parts of the talk, of course. And I hope I can entertain everyone that way. So in case you don't know what is bioluminescence, so here's a definition from Cambridge Dictionary. So it's basically the phenomena of light production by living organisms. You might have seen fireflies or some worms that generate their own light, mostly during the night. So that is basically what biologists call bioluminescence. And if you look at the tree of life, let me explain what this tree of life is. It is basically what Darwin and Haeckel drew a century ago. So each of these circles show millions of years. So life started around 4,000 million years ago, around here, by bacteria on Earth, when chemical materials somehow magically happened to be a cell, which was bacteria, and the bacteria evolve, and then they form archaeas, eukaryotes, and now we are here today, where we humans and mammals exist. So if you look at these, and there are multiple extinctions, as you can see in the circle along the way. And if you look at these different kingdoms of organisms, many of them, the one I'm showing with an arrow, actually feature bioluminescence. So fishes, fungi, eukaryotes that I will talk today, and bacteria, they're all in different species of them. They can actually, by themselves, generate light within themselves. So the study of bioluminescence is extremely important, back to the study of origin of life, in a sense, and also the evolution. So it's a great tool to study evolution. So here I use this beautiful artwork that Eleanor Lutz, former PhD student in Washington, actually made, showing all the organisms that generate light. Some of them are like bacteria or small, some of them are like jellyfish or big, and you can see they actually also generate light in different colors, mostly blue and green, and combination of both. And I will talk a little bit about the colors of bioluminescence today too. So you go from very small small organisms that I show on top to some very big one like shrimps that are a few centimeters, or some squids that are like 20 centimeters, and all the body can generate light. What I will talk about today is this one on top left, the Dinoflagellate. So this is a single cell organism. Some of them are known as sea sparkles, if you know, if you heard that. So bioluminescence happens in land and also in the ocean. In the ocean, the estimation is over 90% of living organisms actually can generate light. So here are some of my favorites. So the cowfish on top left, it can generate light around the spectrum of blue and purple. The one on the bottom is the so-called sea angel, and that also can generate light in blue and yellow spectrums. Some of them are not as pretty. So here are two more examples. The one viperfish, these two are actually extremely important in terms of ecological perspective. Viperfish in particular, because it generates two wavelengths of light, so it's blue and red at the same time. And the reason that they do it is they actually, with the red, they attract a small prey, and small prey see what's happening there, and they eat them. Anglerfish use it for the same technique. So anglerfish has this, it's a female anglerfish, the male is much smaller, and they actually have these lamps on their head. And this lamp is not part of their own body, in fact. So what generates the light are bacteria that are living in the fish and generate the light. So it's a symbiotic life, pretty much at its best. So what I will talk about is actually bioluminescence in the ocean too, but I will talk about this type of bioluminescence. So this is a video captured by family in Jervis Bay. And what they saw, I mean, walking at the shore one night, they saw the whole ocean went blue. And when what is important in particular for my research is, I would like to draw your attention to, is when they put their hands inside water, or when they move their feet at this moment, the ocean starts to glow. So it already hints you as a physicist or engineer that some sort of mechanical stimulation is resulting to light production. Okay, so that was the moment that was also very much triggered by this fact. So the reason this happened is actually because of these unicellular cellular organisms. So these are they're called dinoflagellates. They come in different forms. These needle-shaped likes are called fusiformes. The one on the right that I will talk about them are called Pyrocystis Lunula. All of them generate blue lights. And what happens is around the spring, the weather gets really warm. We also, humans, put a lot more waste into the water. So we basically feed them very well. So they start to grow, and they grow like crazy, and they bloom, in fact, in the ocean, and they are capable of generating light. So the phenomena like what I showed before happens. Okay, so we were not the first people actually who were amazed by the phenomena of bioluminescence. It's one of the oldest philosophical or science topics that humans and mankind start to wonder about. It goes back to 3,000 or 4,000 years ago, apparently, but the first written text seems to be, it goes back to the ancient Greeks. And here in a few slides, that's what I said, that I'm going to do the literature review a little bit unconventionally, going really back to what humans wrote about bioluminescence. This is the first time I'm doing it. I hope it kind of fits the spirit of the journal club. So the first written text about bioluminescence seems to be going back to Aristotle himself. So this is around 2,400 years ago, in his book On the Soul. That is a great read for for scientists and philosophers. I mean, I recently myself read it. So what he writes about, what he saw as dead fishes, because when fish dies, now we know that the bacteria that grow on it can also generate light. So it's some form of bioluminescence. And he says that some objects of sight, which is light, are invisible in darkness, stimulate the sense. That is, things that appear fiery or shining. And he says that this class of object has no simple common names. This is the name that we have now these days called them bioluminescent organisms. It goes through the very rich history of, especially including superstitious, that people thought for years that lights coming off the bodies, especially dead bodies, are the soul leaving the body. I'm not going there, but I jump directly to 1900s, where the very first, what I call systematic study of bioluminescence happens by James McCarthy. He's an Irish naturalist who wrote in Royal Society around 200 years ago about not only the bioluminescence on the microscope. So this was a microscope. I guess it came from Netherlands to now the UK. They perfected it and they went to the ocean and they started looking at microorganisms. He pointed out not only on this mechanical stimulation, this is 200 years ago, but also talk about the shapes of these organisms. As a matter of fact, I will come back to the shapes also towards the end of my talk. In between, the books were also written. This is the first book written by Conradi Genzerny. I mean, he reviewed the literature in ancient Greeks, in India, and also the Arabic literature on what people wrote about bioluminescence, and wrote this book. Unfortunately, I couldn't find the translation. It's all in Latin. But it's basically 300 pages just about bioluminescence. And to us, it's apparently the first time someone wrote it. This is early 16th century that someone wrote fully a book about this topic. Now I'm trying to jump to what the real science, in a sense, started. So centuries before, alchemists were searching for this so-called philosopher's stone. This is a stone that could change materials to gold and silver. This is basically foundation of chemistry and many of parts of physics to some sense. But with the rise of the modern or early modern alchemy, in particular Robert Boyle, people start looking at bioluminescence in a different way. So you probably know about these famous experiments of Boyle, who made this somehow vacuum chamber. And what he did, he actually put bioluminescence organism in the vacuum chamber. He made it with the help of his assistant, Robert Hooke. And what they found was that when they evacuated air, bioluminescence didn't happen. So this was the very first evidence that to generate light in living organism, you need air. And nowadays we know is oxygen, in fact, that you need. So the last historic slide that I want to make goes back to Raphael Dubois, the French naturalist and scientist, that he was the one who basically unlocked the secret of bioluminescence. So he basically found out these two chemicals that he named luciferin and luciferase, and the one an enzyme, one a molecule that generates light. But I recently read a little bit more about his books. What I much liked, beside bioluminescence, was this sentence in his book that he start calling matter protein and living matter bioprotein. And in the end, he basically mentions that basically matter and living matter are the same. So it's a very neat philosophical points that goes somehow maybe connects to the topic of reduction and emergence. So maybe interesting for people here that the both topic of emergence and reduction was a thing when people studied living matter, including the topic of bioluminescence and the origin. So what Dubois found was basically these two. So the left is this molecule called luciferin. And the one right is called luciferase. This is for dinoflagellates. It's a common name, so it's not an exact technical term. So you can have luciferin, luciferase for fireflies or other organisms, jellyfish. But these two are for dinoflagellates if the condition is good, meaning that you have satisfied the low pH, and sometimes you need ATP, sometimes you need some other materials like magnesium, and sometimes you also need another bounding protein that should sit on your molecule. But what happens if this chemical reaction happens? You get light, and this light can be in different basic different wavelength. Typically in the ocean, you're in around blue light wavelength, and on land, you're around green and yellow. What is neat is sometimes you can actually excite by your bioluminescence wavelengths another molecule that generates different lights. And this basically was first seen around 70, 80 years ago, if I'm not mistaken, on jellyfishes. So people who studied jellyfishes in Japan and in Princeton, they found this jellyfish that was blue. They start isolating materials in the lab, and they later also found out the same jellyfish sometimes generate green light. So the reason was that this chemical reaction generates blue light, but they found out this other protein that get excited by blue light and generate green light. And that was basically done by Osamu Shimomura, that you might the name. And this is now very known as green fluorescent protein. And in fact, in 2008, Martin Shalfie, Roger Tien, and Osamu Shimomura were awarded the Nobel Prize in chemistry because of discovery of this protein that is called green fluorescent protein. So green fluorescent protein is a protein in some organism, in some living organism like jellyfishes, that gets a light, typically blue light, and generate this green lights. And this completely revolutionized the biology and oncology. And if you go to any lab these days that works on living organisms with microscopy, you can find an organism that has green fluorescent protein. So it's a gene tag. You can express green fluorescent protein to an organism and let the organism grow and reproduce, and you can still have that gene. So on the left, I have an example that people now very commonly use this protein for cancer therapies and for cancer detection. On the right, you see some mouses that are basically introduced with this green fluorescent protein. So you put them in ultra blue light, they start shining green. Okay, so it took over 2,000 years or more, but finally we have this breakthrough in bioluminescence through all this process of interesting history. So the question is, I'm a physicist, what question I can ask after these thousands of years? The question we ask goes back to this video that I showed you at the beginning, that what triggers bioluminescence, right? So we know a lot about this biochemistry that happens inside the cell, thanks to the biochemist, but we actually know very, very little about the mechanism of triggering this chemical reaction inside the cell. And that's where we start working on the topic since last 2 years. Okay, so what we did, we got this organism. It's called Pyrocystis Lunula. It's called Lunula because of its similarity to moon. But unlike moon, it's completely non-motile, so it doesn't move around. So that makes the life of experimentalists very easy. You can easily study in the lab. A little bit of biological background. They have a very thick cell wall, and then they have this lipid bilayer that we think is where the stimulation occurs. And they have this GDP receptors that I will talk about it later too. They have this core that you wonder what is happening there. The real answer is that there's a lot happening that we don't know. But when we do fluorescent microscopy, you see spots that are, we believe, is mostly chlorophyll that generate autofluorescence in chloroplasts. What is important is that it looks like by mechanical stimulation, we generate some chemicals, most likely calcium, and there are these organelles called syntholones that are very particular organelles that keep all these chemicals, luciferin, luciferase, that I said earlier. And then through some action potential, so by hydrogen proton release, you reduce the pH, you make the environment suitable for this chemical reaction, and then this oxidation of luciferin via the enzyme luciferase happens, and you get light as a response. So basically, you go from extremely small scales like molecules and ions to something that you see by naked eyes. And imagine when you have millions of them all together, they have this collective light generation. So what happens is basically the emergence of these breaking waves in the ocean that is blue. One interesting point that early physicists actually got very interested in is that this light does not generate heat. So this is the so-called cold light, in fact, through this chemical reaction. So some people actually use this bioluminescence during the wartime in Japan for reading. So they got these organisms that could generate light. They were in an environment that they couldn't put fire on. So they use organisms to actually read things or communicate. There's very neat history about World War II and World War I and bioluminescence too. Anyhow, so what we did, we went to the lab. We had the microscope. We started making the setups that we can control in micrometers, manipulate our organisms. We have two pipettes here, as you can see. Let me show you what we've done. So we basically went fishing. So with one of these micropipettes, we're talking about a few micrometers only, right? We start grabbing one of these organisms. So all I'm seeing is on the microscope. So these organisms are around 10 to 50 micrometer length. So what we do, we take one of them. You already saw maybe they generate light, but that's not the main point. I fast forward a little bit. What we do, we actually keep them between two micropipettes. So this is one single cell that I can control between these two pipettes. And now I try to push these, basically try to squeeze these organisms and see if it generates light. Okay. So what I didn't show you is that one of these pipettes is actually like AFM tip bended. So through the deflection of that pipette, I can also measure the force if I want. Okay, so let me show you the video. I'm going to squeeze the algae now, or the cell now, and you can see it began to generate light. So let me do the amplification. So as I squeeze the algae, it starts to generate light. So all this chemical reaction that I told you, basically we stimulate them by mechanical deformation of the cell itself. It happened to be an extremely useful protocol for many other scientists in biophysics, because, for instance, in cancer cells, we also try to understand how cancer cells respond to mechanical forces, and such an assay or bioprotocol can help us understand if I put force on a cell, what's the response of it. It's really good here because the response is light, so we can immediately measure. Okay, so this is basically the picture of the same video I showed you. The algae is squeezed. So this is the amount of deformation that you need, and you can actually get the light. So what was, so basically the first discovery, if I call it, was that membrane deformation was needed for light production. So the way we look at the problem was the way we look at dynamical systems. So I look at the intensity of light in an arbitrary unit as a function of time. And this is each time that I squeeze the cell and I get this signal. So I can plot this in a somehow phase portrait form. So I show intensity on x-axis and the derivative of it with respect to time on y-axis. And I get this loop. So this is my first loop. I generate light and then it decays back to 0. And the second squeeze, and as I go on, the cell runs out of fuel. basically, and it doesn't generate light. So the light decays over time. What was extremely interesting for us to find out was this. So if I get the cell and I basically squeeze it very fast, but to a very small extent, as I'm showing in this video, so you barely probably see the movement of the pipette in the bottom, there is no light production. But at the same time, if I squeeze the cell very largely, but very slowly, the cell doesn't generate light either. Okay, so this made us thinking how cell decides about light production. You should keep in mind, this cell is extremely ancient, so it's not really evolved. This is billions of years old, and there's no neural network, there's nothing. So somehow it gets this ability to respond to the mechanical stimulations when it's important. So what we did, we got this IMAX, we repeated these experiments many times, which was not a very easy job actually, but and then we ended up to this graph. So what I'm showing here in this axis, I'm showing how fast I'm squeezing the cell, and in this axis, I'm showing how much I squeeze the cell, and the z-axis is basically the amount of light I get. Okay, so it looks like the cell only responds or only consumes its fuels when someone is basically punching it fast and hard enough. Okay, so that means because this is a defense mechanism, so they generate light. I forgot to say that they generate light to scare the predators, or basically call for a bigger predator to come and eat the smaller predator. So this is called burglar alarm. Okay, and somehow during the course of evolution, they learned to use this little bit of chemicals that they have inside only if the danger is actually real. Something is moving there really fast and also really a lot in terms of the formation. Okay, so we were very happy with this. The question was, can we present a simple model for this mechanism thing, mathematical model? And the way we did it is, again, looking at it as a dynamical system. So we decided to see if we can write a system of linear differential equations to actually solve for what's happening here. So on the left, the X matrix is basically a stimulation or biochemistry response, like everything goes there. This A matrix is actually what we're looking for, which what I call the constant matrix, and in this case, bioluminescence constant matrix. And this includes all the dynamics in my problem. Okay, it depends on these parameters on the left-hand side, and then there are these external parameters that I put all of them in matrix. Okay, so how does it look like? So I told you about this. This is where also my own questions start. So I put some stress, and I told you I released this chemical, let's call it calcium, and I would like to note that the detail of this mechanism is extremely complicated, and at least to me very unknown. Typically there are these G protein coupled receptors sitting on a membrane. These are folded proteins on the lipid bilayer, and typically what is known, and if a chemical signal comes, a ligand comes, they start to activate something inside through this GTP hybridization, and then the signaling inside the cell happens. This is very key for us. We don't know the details of it, and we would like to know more. Another things that can happen with G protein coupled receptors is that they might actuate ion channels. So you can imagine there's an ion channel also here on the membrane, and that results to the opening of these channels and the release of ions inside the cells. We really know little about that, but what we saw in the experiment, it looks like as a response to our mechanical force, there is some sort of viscoelastic response, meaning that it depends on the deformation and the deformation rate. So the easier thing I could think of, literally I couldn't get easier than that, was what if I use a Maxwell model as for my stimulation system. So S, you can think of it as amount of chemical component released by this mechanism via the stimulation delta. Okay, And of course, there's a time scale in delta dot, how fast I deform the cell, but there's also this time scale of tau e that you can call it a relaxation time, if you want to keep the language of Newtonian mechanics. Okay, in the lab, our protocol looks like this. So delta dot was fixed for a given time, and then this is a linear equation, you can solve it, and your S looks like an exponential decaying function. And you can get how much chemical you release by taking the integral over this time of your release signal. Okay?
Sorry, Mazi, just
that's yes.
Sorry, your delta there is what exactly? It's a deformation you apply.
Exactly. So delta dot is a deformation rate.
Okay.
And delta is how much I deform. Exactly. So this is my stimulation of the viscoelastic membrane, let's call it. I need more differential equations, right, to model the chemistry inside my cell, which we call it hidden biochemistry. So I ignore all the details, what happens inside the cell, and I coarse grain everything to this extremely simple differential equation that is linear with respect to H and S. So H is, again, my hidden biochemistry, and S comes from the first equation by the stimulations, and then I have another linear differential equation for the light, that basically I is the amount of my light production that is, again, linearly negatively depends on H and I. So I now introduce two more time scale, tau A and tau R, in my systems, and we hope that by coupling this equations, I can actually model what I saw in the experiment. So in the language of equations I showed earlier, I know my X vector is S, H, and I, and my constant matrix looks like this. And now I only have one component in my stimulation
Mazi,
vector.
Can I ask a question? Just sorry to interrupt, but I mean, can you actually be more specific about what H is, except for just hidden? I mean, is there also a meaning to it, in a sense?
Yes. Yeah. So the meaning is basically is all the chemical reaction happening due to. So basically, the more S I produce, basically the more H I produce. So it's basically this. I basically coarse grain whatever happens between calcium production, the action potential, and the chemical reaction itself. So we could do actually better. So what we could do, we could get all these chemical reactions that happens inside the cell after the signaling is started. And for each chemical reaction, we could write one differential equation coupled to each other. And instead of doing that, I decided to actually put them all in one differential equation. So this is
is then the main function that there is some sort of a delay between S and some later reactions? Because you could, of course, that's hidden. Yeah, sure.
Right?
Okay, but so it's a delay function basically. Then.
Exactly.
Right.
You can look at it as delay function.
Yeah.
Exactly.
Okay. Thanks. That makes sense. Thanks.
So maybe along the same lines, maybe I didn't follow everything, but again with this hidden biochemistry. So you said there's all these reactions that are being coarse-grained into this equation. Can you actually write it like that? That if you write all the reactions, then you can kind of like pile everything into H that is equal to some other combination of stuff, and this is really the equation that you get, or there's some other approximation in between?
So I didn't do it that way, so I have to think. But I think you can. The reason being that when you look at this chemical reaction, I mean, the way you write it, you typically write a linear ODE for all of them, right? And what happens is that you add more and more constants to them in the end. I think you can, but I have to think actually more to basically say how rigorously you come from all the sets of differential equations to this one hidden one. But I assume you can actually. Yeah. Okay. So this is the set of equation. You can solve everything because everything is linear. Here's the answer. So I looks like this is a combination of bunch of exponential functions. Okay, but it's easier to look at the results. So this is the phase portrait I told you for multiple experiments. And I didn't show all the experiments, but they all look like this. And the line, the white line is basically our theoretical line where you are solving these differential equations. Okay, for I in particular, it fits very well. And interestingly enough, we found out there is a small dependency on the constant times, in fact, for all these experiments. By variation of deformation, by variation of deformation rate, and also cell-to-cell variation, which is very rare in biology if you look at the experimental biology papers. So that means we can now construct this bioluminescence, what I call bioluminescence constant matrix, meaning that for these particular organisms, we now know how long it takes for the light to rise up. We know how long for the rays to decay. And this is what this matrix A means. And the good thing about it is that we can now compare it with different organisms that are, for instance, evolving different stage of evolution, or they belong to different kingdom, and compare them basically via numbers. So it's basically some quantitative biology that it seems to be very useful.
Wait a minute, Mazi. Can I ask another question? I mean, this biology and number statement, and especially about the numbers themselves. I mean, what are they supposed to be? I mean, they're time scales, no?
Yes.
And so how do I interpret this? Is this a 33 milliseconds or microseconds or something?
Okay, yes, so this is all milliseconds. That's a great question. So if I go back to the definition of A, so each of them are basically these few numbers that they have, time constants that they have. For instance, 1 over tau e being the relaxation time of the membrane. So if I go back to A, which I guess it was minus 33, so tau e is basically in seconds, give me the elastic relaxation time of the membrane. Same with tau A and tau R. So I guess tau A was around few also milliseconds. Same with tau R, which is longer. So tau A corresponds to the time for the signal that rises. So if I go back to one of these curves I showed, for instance, here. So tau A is the time that corresponds to the signal to after a stimulation to reach its maximum, in a sense. And tau R is the time from this maximum decaying to zero again, because light goes up and comes down, and then it goes up and down in different time scales. And that's what tau A means and what tau R means. They're all around milliseconds, including tau, the relaxation. Sorry, I had to mention that.
So in your sorry, in your model, you replace those tau A and tau R that you measured on the experiments, or did you fit them from the final? So you replace them from the experiment. Sorry. Yeah.
Sorry, yeah, but we fit everything to the experiment. So that was an interesting point I wanted to try to highlight. So if you plot everything from your experiments in such a curve that you normalize your I by I max, and you normalize I dot by I dot max, so basically they are self-similar in shape. So no matter what cell you take, and no matter how many times you squeeze them or how you squeeze them, the shape of your signal never change. And that gave us this opportunity to do this fitting. So we got somehow lucky. If cell-to-cell variation was a lot, or cell decided if it was a non-linear viscoelastic response, then we had to do something better than this. But since a system of ODE could do it, we just stopped there.
So the A here is fitted from this graph here, and
yes.
and from that all your elements in the matrix, which are now integers. So I guess you fit it in a certain way that you get integers. So all your elements in the matrix now they matches to this time scale. So what you're saying is that luckily all your I mean, how you say, all your experiments here, they had the same tau E, tau R, tau A, and I don't remember what exactly is the last one. I see.
Try to see if I can find the curve that I had. I mean, maybe in the end I can show you, but if I plug, yeah, exactly. So if I get one of these curves that I showed earlier, so one of these, for instance, the first peak that I have here, go up and down. So we have around 300 plus, I can see if I remember, 360 of these experiments. And if I put all these 360 experiments on top of each other, they actually look alike if I normalize it with the Imax and I.Max. And that's exactly, that allows us, in fact, to do what we've done, which was fitting through this type of curve.
Is this a property of the type of single cells that you're using? I mean, are they somehow all alike, or I mean,
we don't know. So that's exactly, yeah, we don't know. So that's what I think this modeling and this matrix, although very simple, it can be very useful. So we now growing two other type of cells, also dinoflagellates, in the lab, and we try to actually find if these numbers vary. They are different in terms of evolutionary heritage, but they are the same family in a sense. There are different shapes. So, so now we have this opportunity to, via this simple model, to compare different organisms. So that we don't know.
Mazi, can I ask one more question?
Yes, please.
So, I mean, I see these numbers and they look terribly exact for anything that is fitted somehow. So is this, were these rounded up, or is this, or I guess, how was the fitting done to get this?
No, it's rounded up for just for the sake of presentation. The exact numbers or something else. Yeah. But close to this, and there is, of course, there is a standard deviation, right? I mean, between that, again, it's just for the sake of presentation. But to give you a taste of how big tau are and how tau A.
Yeah, just for my understanding, these numbers are in fact 1 over the time scale, so they're rates.
Yes.
But it looks like the middle one was the activation, no?
The middle one is tau A, in fact, yes.
Yeah, but so that's smaller than tau R. But exactly. I mean, in your curve, it's the other way around, not? It's the activation which is faster than the relaxation.
They go rather fast up. So tau A is the basically time scale from 0 to here.
Yeah,
and tau R is the basically time scale going from here.
And then the relaxation is slow. So I'm having trouble understanding these numbers. I mean, I would expect the other way around, actually. But okay, this is just
maybe I understood well, actually.
Okay, but this is just you probably can solve this probably very easy.
So yeah, so tau R is the
could it be that
yes,
you're
indeed. Yeah, no. Yeah, you're right, actually. Yeah. So it was the other way around, they said. Exactly.
Okay. So you're reporting tau here, you mean?
Okay. Exactly. Yeah. So we're just reporting time scales. In fact.
Okay.
Yes.
Okay.
Yes.
That's clear.
Yeah. Okay. So so go ahead. I mean, I showed you in the beginning some phenomena that was driven by fluid flow, and that's where actually my main expertise is. And we basically decided to also see if we can stimulate the cell with the fluid flow. So we did what we did in the lab. We got again one of these cells. We got another pipette, and instead of squeezing it this time, we start generating flow over it by flowing fluid around it. And the particles are basically there to just show you how the flow looks like. So I can zoom in a little bit, and I'm shooting the fluid here, and these particles, you can see that we can track them, and by that we can actually visualize the flow around the cells. So this is another protocol that we try to develop by this time stimulation of flow. So this is my cell. Now I remove the particles to avoid any biological interference. The flow is coming down from the pipette on top. Dash dot is the initial position of the cell. And as you can see, as I push the fluid to the cell starts to deform. And when the deformation is large enough, it starts to generate the light. Okay. So the question was, can we also get more information in terms of modeling this, because again, if we could make a biological assay that I go to the lab and I push fluids on the cell and I look at the response, in this case, light production, then I can also I basically have a very useful mechanical sensing assay. So here you can do a lot of cool stuff in the lab. You can visualize the flow using particle image velocimetry. You cross-correlate images, and you get the full velocity field. You can do all the particle trackings and all the things that are common in hydrodynamics. And of course, you can also capture this light production. The problem that we faced was that we did this, but we couldn't actually find out what's the pressure on a cell, right? So because the problem is you cannot put a pressure sensor on a 10 micrometer cell in the lab. So we went back to the theory and we said, can I model such a system? So my system has an elastic body, my cell here with some curvature R. And then I have a jet that is blowing liquids to the cell and deforming that. And the question was, can I measure force or pressure on this solid body? So as one typically does in this situation, you go back to London Lifshitz and you find out some relevant example or exercise. So what I called Zaleskin-London square jet, more commonly known as London square jet, is a very good model to start modeling this. So let me explain what is it. You basically say I have a Stokes flow. So I have continuity equations and I have the momentum equations. So this is Navier-Stokes for the limit where inertia is not important. And I replace my jet with a singularity. So the singularity is where the flow is generated with the power F naught. Okay, so I basically solve the Stokes equation with the Stokeslet in a sense in it. And then I have boundary conditions. This is all without this this cell in front of it, and that's been done again a few decades ago by Lambda Square. And what we've done, we actually put now this elastic body in front of it, and we try to solve for the pressure. Okay, for the first order terms were already known for a flat surface by Blake. What we've done first, we start putting deformed surfaces and solve high order terms that look at this. And more recently, we also made it fully elastic and we coupled liquids and elasticity all together. And again, to put it in a nutshell to what we've done, we can now, using this simple modeling, look for pressure on the cell and the forces exerted on the cell and couple it with our experiment. There is one neat but very technical difficulties here, and that's this length scale A that is in my problem. So in reality, I have a jet that is generated by this nozzle has a diameter A. But in my singularity problem that I showed earlier, there's no length scale, basically. So as A goes to 0, I go to a very singular point. And the question is, how can I match reality or experiments with such an analysis? So we did that via solving the full problem numerically. So this time we solved the whole Navier-Stokes equation. It's full glory. So that's this equation, continuity equation, momentum equations, and this is the stress equation. We couple it with elasticity, with the hyperelastic solids. So that is governed. So now US is the deformation of it, and the gradient of the sigma S is basically your stress gradient. And these basically depends on the model of elastic material that we use, and we got a model available. We use finite element, a commercial package in the end, to solve this problem. Here is my jet. On the right, I'm showing you the pressure variation, the log 10 of it, so it's more clear. On the left, I'm showing the streamline of the flow and how this sheet, the cell wall of my cell, deforms due to this. I don't go to the technical details, but in a nutshell, what happens. Oops. is, so we've done this Stokes-like analysis. There was this awkward business with this finite length scale of my jet that we had to do some numerical simulations to get this table. So this is a readout table now that you can find this constant that compensate for this lack of length scale in my theory. We've done it already, so one can complete this biomechanosensing assay. What does it mean? If now someone has a cell in the lab and wants to see how does it respond to fluid, you can do what we propose to do. You can push fluids on it, and you can come back to our analysis. We now have a big equations, and depending on your cell elastic structure, which is hidden in this Erickson number, and your experimental protocol, you can read out what's the pressure on your cell, and you see how my cell generates, I don't know, electrons at this pressure. Okay, so we ended up proposing this mechanosensing course. Jay, how much time we have?
About 10 minutes.
10 minutes, okay.
Or a bit more.
Right. So then, so in the last 10 minutes, then let me tell you what we're doing now, actually. So where this problem took us. Something that during the studying these organisms, we found out was that it not only generates light, it also does another creative thing. We found out the shape of the, not the shape, but the internal structure of the cell varies significantly during day and night. And later we found out cell internal structure changes as a response to mechanical stimulation, as a result to light, and a lot of things. So these cells, being around for billions of years, learn how to adopt internal cellular structures to resist, basically, to survive. So what you see from left to right is this is the cell during the day, so when it gets a lot of sunshine, and this is the cell at night when there's not much sunshine. So what it does, it contracts all these chloroplasts inside of it, coming all the way to the center, and that apparently helps to generate more light during the night, because they only generate light actually during the night, because that's the only time that it makes sense to generate light. Okay, so that triggered us quite a lot. So we went to the literature. We found that biologists are actually looking at the problem of chloroplasts, less in marine organisms, but a lot in plant cells, because that's heavily related to photosynthesis. And so here I'm showing the chloroplast movement inside the plant cell called Elodia, which we are also studying now in the lab. And what you can observe immediately is that the motion of them sometimes is way too fast to be considered as diffusion. So this is not diffusion. This is the non-active diffusion. So something inside the cell is taking this chloroplast and move them around, in fact. And you might be familiar with the concept of molecular motors. So there are each of our cells, our human bodies, and also other organisms have these very special motors, the machines that move things around. This famous video is one of my all-time favorite videos. So this is a microtubule, and a molecular motor is walking on it and carrying this organelle around. So in our case, we assume maybe the chloroplast is moving actually like this. So there's a molecular motor taking the chloroplast, move them around. The first time I saw this, I thought this is just a human imagination. This can't be real, but there's now actually experimental direct observation that they literally take this cargo and walk on their highway. It could be microtubules or actin filaments. Okay, I mean, one can model such as systems. I mean, here you can tag them with fluorescent proteins that I also talked earlier, or other dyes, and you see them. They actually process this one. I'm tracking walking on its own road. If you want to go to a single particle level, you can use a Langevin type of equations to model the motion of such organelle movement inside the cell. We've done it. It's very easy. I mean, you get such an equation, an ODE again, and you try to solve it for a proper energy landscape in your system. Okay, and this energy landscape, there are many models nowadays. Theoreticians really love this type of equations, between 25 years ago until recently, and they model a lot of different types of W's, but they all ended up to similar type of equation. You can also go all the way to Fokker-Planck equation. That's something we are considering doing, and now I can search for the probability of where my chloroplast is as a function of different mechanical, light, or other environmental stimulation. So here, for instance, if I take an average of chloroplasts over this line, this is how they distribute over day, and during night they distribute it differently. So, but this is just a way. I mean, we know we should do better than this, but this is an easy way to again model what happens if you stimulate your cells. This is what we're doing now in the lab to basically bring it back to numbers, in fact, in terms of math. Okay, so this sounds very easy, and it is, in fact, very oversimplified. The reason being is life in general and the study of living matter is very more complicated. So here is an example of this chloroplast moving in a plant cells. The green filaments are basically filaments that move inside the cell. So this system is not really one individual molecule or thing moving around that I can model it. So simply the whole environment is extremely crowded. At the same time, everything is very active, and there are this huge problem with the spatiotemporal noises, that you cannot ignore noises in such biological systems at all. And plus all the complexity comes from environmental effects and cells. Okay, so I try to conclude. I talk about these things. There are a few things, very small things that we now know. We now know that the bioluminescence depends on flash intensity. The flash intensity depends on the formation rate. And we have these very simple viscoelastic models to model this. All the things I talked about, that is actually in this PRL paper. You can have a look. There are way more things that we actually don't know. That being mechanosensing in particular. So we are searching for collaborators, in fact, if we could do some molecular dynamics to understand what really happens to lipid bilayer and proteins inside lipid bilayer when we deform it, when we put some force on it. There is all the theory of fluidization of lipid bilayer. I mean, it's very hard for me to grasp it, to be honest. But I think there's infinite amount of work can be done there. Another thing that is very curious for us, and we hope we can work on that soon, is the shape of these cells. So there are a lot of weird shapes of organisms, single cell organism. This one is like, like croissant. There are others with, I don't know, spikes. There are all sorts of shapes, and the question is why on earth they made these shapes. There must be some reason. I mean, at least biology thinks there's always a reason in evolutionary evolution. So I'm a hydrodynamicist, and we try to answer if there is a hydrodynamic reason for these shapes. We have some interesting results already that maybe the shape is the benefit of staying more in the fluids when they're doing this vertical motion in the ocean, but we have to confirm. So we want to study how cell shape affect the hydrodynamics around it, and maybe inverse boundary methods and those type of numerical simulations can help. Okay, I thank my colleagues. Nico was my intern in Cambridge at that time. He's now my PhD student, working on similar problems of chloroplasts now. Michael, Alpen, Ellen, Christoph, Eric, and Ray were colleagues in Cambridge. All of them. Ray was my first postdoc, second postdoc advisor. And fundings, we'll end the topic, my talk. Actually, I talk a lot about history. Something about bioluminescence, a lot of artists also got involved with bioluminescence. Poets in particular, Shakespeare wrote a lot about bioluminescence. I'm not going to talk about poetry, but I'm going to talk about the Dutch painter, Escher, who is very known for his works on because of symmetry, especially from last year and the Nobel Prize in Physics, and Roger Penrose's connection to him. But he also was very obsessed with the bioluminescence, in fact. So he drew ocean bioluminescence. So what he tried to draw here was such a phenomenon when the ocean waves goes fully blue. And he has another painting that blew my mind, but it's this one. He drew dolphins swimming in the ocean while bioluminescence bloom happens. So there's a picture on the right, the color one, dolphins moving through this dinoflagellate bioluminescence, generating light production, and that's an Escher expression. So I end here my talk. I would love to take more questions.
Thanks a lot for this nice talk. Does anyone have any questions?
Silence.
So maybe I can ask something. I mean, it's more. I want to wait for it because I guess it's not a specific question. But so I mean, I noticed that you saw that you mentioned that you're interested in the MD models part. So I guess the question would be, I mean, it seems that the timescales of this bioluminescence is really on the seconds timescales, and I mean you can see it, right? We can really see it on our lifetime. So I mean, MD, you can really do things very, very limited in time in some sense. So my question is, are you interested just in the mechanisms that do the whole thing? Because in that case, maybe a more coarse-grained model is better at the particle-based chemical reaction level, but maybe not at the MD level. So I guess my question is, do you know which part are you exactly interested from the MD perspective?
Yeah, the very first part, not the whole system. Indeed, I mean, I guess the whole bioluminescence and light production is way too much for MD. Not only the time, but it's also way too many things, and also space. I mean, it's very big, right? So these are, I mean, even one organelle that generates lights is, I guess, around 3-4 micrometer, which I think is pushing the limit of MD quite a lot. So what will be very interesting for us is as simple as, even without proteins on the lipid bilayer, I barely can find works that have a lipid bilayer on the shear forces, for instance. How does it change the conformation of the lipid bilayer? How does it really fluidize it? And if it does, if there is a protein on it, does it deform in a sense? And basically the combination of force and foldings, protein foldings, is something that we are interested in. I'm not sure if that's actually accessible with MD these days or not. I'm not an MD expert.
Me neither, but just yeah, I was curious because I guess I'm more interested in the bit coarser grain scale at the more chemical reaction level, but still at the particle base. So somewhere in between ODEs and MD, and that's like the area that I know more about. So just checking what you were actually looking for.
Yeah, I guess with those coarse grain models, one can go. I remember your talk actually here. I guess one can go maybe towards more these light production organelles themselves.
Yeah, that would be the idea. So maybe do some kind of model that it's a bit more detailed than your ODE version, but maybe not as hardcore as going full MD on the whole process. Could be interesting. Maybe there's some additional stuff to be seen.
Yeah, maybe we talk about that. I mean, whatever you do is more detailed than my ODE.
Yeah, but I mean, sometimes it doesn't show anything new, right? So then maybe it's not worth it. But it could be that there's some other stuff. So I guess also depends on what's observed at the experimental level. And if there's some other phenomena that is not being explained by the current models, maybe it's worth delving a bit deeper.
I see. No. Let me actually think about it. That's a very good suggestion. I mean, maybe when it comes to the light organelle, I really don't think people know much about it at all. I think the chemical reaction are known. But that would be interesting. Let me read a little bit more about it and come back to you, Mazi.
Well, if you're interested, I mean, that's more what I do. So if you're interested, feel free to just email me. And then, I mean, I like the problem, so it looks very interesting. So
For sure.
I wouldn't mind trying some things out.
Absolutely, for sure.
Can I ask you a sort of maybe it's a very busy question, but since you've shown these pictures of the waves and the paintings, so is it trivial to model just that kind of ocean fluid with luminescent stuff in it?
To model the flow, the waves.
Yeah, with that that sort of large scale fluid with luminescent stuff on it.
Yes, yes. So we have a bachelor students trying to do that in the lab now. So what she's doing, she basically has a tank, and we move the tank left and right, and we generate wave in it, and then we introduce the algae in it. So the idea is if we can actually track use the algae as a pressure sensor in the waves, because I can solve Navier-Stokes in two phase and get the wave, and then
but is the model just it would just be some sort of a fluid that is that carries some charge, and that charge is some particle concentration of the algae, and that's it, or there's more to it than that?
It's more to it. So it's a flow that generates shear stress inside the body, inside the flow domain. And if the stress inside the domain is above the stress that generates light, it generates basically it results to light production. So you can solve a Navier-Stokes plus active reaction, diffusion, convection equation.
I see.
And the reaction part comes from that is coupled with the stress tensor in the fluid, or the shear stress inside the fluid.
I see. So have you calculated something like modes, like sun modes or whatever, propagation modes, and just wave profiles, like sun speed?
No, we haven't. But I see where you're going. I guess you're thinking about some collective motion or collective behavior in a larger scale.
Yeah, right? Exactly. Yeah, that's what I'm thinking.
Yeah, there's more interesting. So these algae, they don't talk to each other. So we tested that. So the light of one algae doesn't result to the light production of the other algae.
Okay.
So that was a huge disappointment, because then we could talk about this bigger emergent behavior.
Yeah.
But there are in a multicellular organisms like jellyfish. If you look at the bioluminescence of jellyfish, you see waves. So you basically one neuron starts to generate light, and the other neuron triggers, and they go in a round. Mm-hmm. It's extremely complicated and way more beautiful than one single cell generating light. That's something we have planned to do, but it's really hard to do experiment with these jellyfish. Mm-hmm. So what people do, our plan is to collaborate with oceanographers who do take pictures of these organisms in nature, and then using their videos to just do processing and then learn about biology and come up with some wave modeling. But in the ocean, they just respond. They're very smart, but they're also very stupid. They're one cell, right? So yeah. they just respond to the ocean waves.
Mm-hmm.
Yeah.
I see. But that coupling is trivial. Like understanding that coupling is.
I think so. That coupling is trivial if your flow is trivial, like simple waves. If you have dolphins or submarines, then things get more complicated, because the flow gets nonlinear, and then turbulence might come in.
Right, but just the normal waves. I mean, that you have an easy model for that. Yeah.
We don't have it yet, but we can model waves by solving Navier-Stokes, I guess, but yeah. Not mathematically. I guess if you're thinking about a fully mathematical equation, waves get a stress below it and couple it. No, we don't have a model for that.
Okay.
It would be interesting actually to do that.
Okay.
So, yeah.
Okay. Thanks. Does anyone have any other question? No, then I guess we should thank Mazi again. Thanks a lot.